Hello, thank you for the opportunity to present. My name is Doug Densmore. Unfortunately, I won't be able to stay for the Q&A section, but my postdoc Samuel will. I'm here to speak to you guys about the vision of the DAMP lab, about how we can make remote services, workflows, and infrastructure available for synthetic biology. So my lab, what it does is take concepts from electrical engineering and apply them to the creation of biological systems. It makes tools like Cello that take high-level descriptions and compile them to DNA circuits. Eugene, a programming language which describes the way in which synthetic biological circuits and pieces can be composed. Tools like Raven that design assembly plans for those DNA segments. And then tools like Puppeteer, which then plan how liquid handling robots can put those pieces of DNA together. We also make microfluidics that can then house and then test and assay those biological constructs. And we can put them together in a specified design, build, test cycle, where the output of one becomes the input to the next, and we can create a whole end-to-end workflow. But when you've done this in silico, the question then becomes, how are you actually going to fabricate these in the real world? Or how are you actually going to test these in the real world, particularly if you only have a computer science background or do not have an experimental lab? Well, the idea is this would go to the cloud, but the biological cloud. And the biological cloud could be something like the design, automation, manufacturing, and prototyping lab, where designs created in silico could then be fabricated and tested at a distributed set of manufacturing facilities that are all doing things in a replicable, automated way to provide services democratized across the entire community. So this is the Center for Integrated Life Science and Engineering at Boston University. On the 4th and 5th floor is the Biological Design Center. This is where the DAMP lab physically is. It has two components. One is the north side. The north side has a relatively complete set of low throughput, heterogeneous processes that personnel run in a very standardized way. And then historically, what was called the south side has large scale automated facilities, or that's what it aspires to have, that then do relatively homogeneous sets of services, but do them in a high throughput, low personnel way. So it has two aspects, the DAMP lab. One is a research vision, where we do lots of synthetic biology research, both in design, build, test cycle, and also the manufacture of microfluidics to test and house these systems. And then we then transition those research aspects into services that external and internal people can use. And we do this with a focus on automation, reproducibility, standards, high throughput, data exchange, cost, and then quantitative metrics. And I want to draw your attention to two of these. One is reproducibility, the other is metrics. We want to really measure and quantify how well we can do these designs, and we want to do them in a very repeatable way. What I do on Monday, I want to be able to do on Friday, and what I do in my lab, I want you to be able to do in your lab. All of the automation in the world is not going to be very helpful if we can't measure how well it's being done and if we can't reproduce it. So we're a core facility on the Charles River campus of BU. What that allows us to do is offer services. If you were to navigate to these websites, you could find our services and you can find the costs. We also do this in a compliance with the International Gene Synthesis Consortia. What this says is that we can do our operations in a way that's responsible and also prevents the creation of any biologically hazardous materials. The services we create are related to automation, high throughput DNA assembly and cloning. We also do things like mini prep and glycerol stock preparation. We purify DNA, we do mRNA enrichment. We do fluorescence-based assays. We do materials storage and basically can house things for researchers that don't have that capability. And we can also do transcriptomics. The idea is that you sit down at a computational tool. It could be one of ours or a tool that you use. You produce DNA designs. Typically, these are collections of pieces of DNA that you want to put together, for example. Then you then send those designs in silico to us, where we design one of two types of plans. One is a automated set of instructions for liquid handling robots. The other could be a collection of protocols created in the environment called Aquarium from the University of Washington, where manual steps be carried out in a very replicable, standardized way by human technicians. So technicians either go through a series of steps or liquid handling robotics do. We have Samuel, who runs the research side. Ali is a microfluidics manager, so manages the fabrication. We have computational experimental individuals, and then we also mentor undergraduates and do lots of education and training in this space as well. So McDonald's operates by making many menu items with a relatively fixed and small set of ingredients, And then those sets of ingredients are put together with technicians in a highly replicable way, in a very standardized and high throughput manner. Similarly, our menu, so to speak, has 38-ish protocols made from a relatively finite set of services and techniques. Then technicians can go about doing this in a very replicable way, relatively high throughput way, standardized and tracked. And also the technicians can have a standardized training regimen by which we can convert undergraduates into automated laboratory technicians. We can also do this in a more automated robotic way with large-scale liquid handling robots like those from Hamilton, as well as smaller, lower cost liquid handling robots like those from Opentrons. We also have desktop milled CNC machines that our lab can use for microfluidic fabrication. On the cloning side, we can do about 6,000 cloning operations in a 24-hour span. With a low-cost robot, with a higher cost robot, we could do almost 13,000. We can assemble hundreds of thousands of fragments with an average cost of reaction of about $1.50, cost per plate of around $140. We can also think of the average cost of those operations I presented in that table, about $22, and we can do anywhere between 24 to 38 different molecular biology protocols. We can also fabricate low-cost desktop milled CNC microfluidics with a feature size of around 75 microns, and also a fabrication time of less than an hour, depending on the design size. You can see all the services that we provide in a dashboard. You can see them being tracked as they progress through. You can check the status. You can then find out what's queued up next. You can see the status of the equipment. There are also biosecurity checks that go on in here, so we can make sure that the people submitting jobs are who they say they are, and we can also figure out what they're being made and make sure we're doing things in a responsible way. We do things like what's shown on this slide, which are creating genetic circuits. This is just an example. The graph on the right is showing all the different steps that would need to go into making these circuits, just to give you some sense of the scale. But also you can start to find parallelism in this graph. How many people would need to be used, liquid handling robotics, what can be done in parallel, what needs to be sequential. We can think about scheduling this. This is an example of a hierarchical assembly. So we would be putting together modular pieces of DNA. This process is very hard to do manually in terms of both the actual experimental work, but also the infrastructure to track these samples, store them, figure out what you have done and what you haven't done. Software and automation helps with all of this. We also can do services like RNASeq. So not only cloning, but we can also think about assays and ways to test if what we made does what we think it does. Testing for COVID, big issue. So what BU did is we converted much of the DAMP lab south into the BU clinical testing facility where we do RT-qPCR tests. We do about 6,000 of these tests a day. We brought in 8 liquid handling robots and 5 qPCR machines. We then program them such in a way that a series of intake samples go through a relatively standardized process using these robots and technicians to provide, again, up to 6,000 tests a day. This is how BU has operated and how BU has been able to stay open and provide classes during all of this time. You can see the results at a real dashboard available to the whole community. So the question for this audience is really, what services are needed for materials, fuels, and chemicals? What services are unique, and how can we parameterize those? Which could be shared? Think about the scaling and economic factors in this space. How could an organization like the DAMP Lab help? What is the role for a distributed collection of biofoundries? How could other folks doing the same work be used to come up with a standardized method that can be used across the whole industry? And what are the automation gaps that exist in these space? Like, which ones should we be addressing, and how can we help with all of these efforts? So again, thank you very much. I appreciated speaking, and I look forward to hearing about the questions you all had for me. Thank you very much.